Hi. In this video, as you can see, we'll be talking about cell membranes and transport with a little bit of diffusion thrown in there. This is part of the second unit of AP Biology. Cell membranes are super important because life needs a body. The cell membrane is the main structural component of a cell, and a cell is the main structural component of living things, so a cell membrane is pretty important. It needs to keep the outside stuff out and to keep the inside stuff in, which another way of saying this is that the cell membrane needs to regulate the movement of materials coming into and out of the cell, right? Life needs to be controlled. We don't want things to just go wherever they want to go. And so a cell membrane is a good way to start going about doing that. The function of the cell membrane is, of course, based on the properties of the phospholipids with their phosphate heads and fatty acid tails, the hydrophilic heads and hydrophobic tails, which is going to make this an interesting molecule because part of the molecule likes water and part of it hates water. And life takes place inside of water. And so when you put a bunch of phospholipids into water, the tails are going to be unhappy and they're going to try to get away from the water as best they can. And the best way to do that is just surround themselves with other hydrophobic molecules. So they get a line like this, but this line isn't enough because there's still the bottom part that has water around it. And so if you get two lines, a phospholipid bilayer, the water kind of gets pushed out from the center and you have this mostly water-free area that is going to serve as a barrier, right? Since most molecules need water to help them to float around and transport themselves from place to place, when you get to this part where there's mostly no water, that's going to be hard to get across. The idea of letting some things in and others not is called semi-permeability. Often said a selectively permeable membrane is another way to describe it. The things that can get in are the things that are small enough to fit between the phospholipid molecules, because remember, those phospholipids aren't actually connected to each other. They're only held together by the hydrophobic interaction, so you can kind of push them out of the way. You have to be nonpolar to pass through that center area, so small nonpolar molecules can pass through. But if you're charged, then you're not going to be able to pass through that center area of the phospholipid bilayer. And of course, no matter what your charge, if you're very large, you probably aren't going to be able to squeeze between the phospholipid molecules. And trying to get a large molecule through is going to end up causing a lot of structural damage to the bilayer. And so on the right, you have this graphic that shows the glucose, which is a polar molecule and relatively large that cannot pass through the membrane. Water is the weird one that water can pass through, even though it is polar, it is very small and it is everywhere. And so they can kind of squeeze their way between, but not very easily. We would say that the bilayer is leaky to water. It's not just like water flows through it super easily, but it can pass through. Now, if you want to do anything other than have a fence, which is basically what the cell membrane is about, then you need proteins to do those. Remember, proteins are the biological molecules that do stuff. And so the most important ones that we would talk about are called integral proteins or transmembrane proteins. They're ones that are inside the bilayer, usually spanning from one side to the other. That's the transmembrane part, which is going to allow us to do a lot of different things. There are some called peripheral proteins that I doubt that you would have to know exactly that word, but sometimes the proteins are just stuck to the very outside of the layer and not inside of the layer. They are stuck there thanks to hydrophobic interactions. The same thing that holds all the phospholipids together also holds the proteins into that. Of course, the proteins fold in a very particular way based on their structure and everything like that. And so it will fold so you get two places that are okay with water and then this big hydrophobic area in the center. And of course, the best place for that hydrophobic region to be found would be amongst other hydrophobic molecules. And that's where it's just going to stay right there because moving from that spot is going to result in it being unhappily next to some water molecules. These proteins do all kinds of jobs. Again, this one is mostly about transport. which are basically tunnels and pumps. But we have a whole section about signal receptors that we talked about. Of course, enzymes are super important throughout biology. Right at the moment, cell-to-cell recognition is important because these glycoproteins are some of the things that your immune system uses to recognize cells. And if you have the wrong glycoprotein on the outside, maybe your immune system will attack it. This is also the types of proteins that viruses use to trick the cell into letting it in. So again, that's kind of interesting happening at the moment. But again, the point is that there's all kinds of different things that you might find on the cell membrane, depending on what the job that needs to be done is. The proteins help us to know that the membrane is fluid, that everything is kind of just floating because nothing's really connected. They took the mouse and the human cell, and they fused them together. And if the membrane wasn't fluid, if it was rigid, you would end up getting kind of a half and half thing, where this side of the final cell would have been all the mouse part, and this would have been all the human part. But because the mouse and the human proteins all kind of started to mix together, then this would mean that they have the ability to float around and move around in the membrane. That fluidity is helped out by the types of phospholipids. We talked about before how unsaturated fats, unsaturated fatty acids are going to be farther apart and less solid. They're going to be more liquid. And then the saturated fats are more solid. And so by changing the composition of the phospholipids in your membrane, you can adjust the fluidity of your membrane. Cholesterol is an interesting one. Cholesterol is one of those molecules that, along with steroid hormones, are large, but they are non-polar. They're not fats, but they are made of hydrocarbons. And you can see in this picture all the cholesterols. They all have these hydrocarbon rings that are just carbon and hydrogen over and over and over again. And that makes them non-polar, and that lets them integrate into the membrane and keep it less fluid or more fluid depending on the situation. In general, the idea would be that cholesterol makes the membrane fluid, just in general. Right, so there you have your fluid mosaic model of the cell membrane, the phospholipids with a bunch of other things, be it cholesterol or proteins floating inside of it. Now, again, the main job that we're talking about here of the cell membrane is to transport things in and out. And there's two basic types of transport. There's passive transport, which uses no energy, and active transport, which does use energy. This is basically based on the idea of diffusion, which is why we'll talk about that in a moment. But it goes about the concentration gradient that molecules are going to want to go from high to low. And so if you just let them do that, then they will use no energy and diffuse from high to low, no problem. But the active part is really the important part for living organisms, in that we don't always want things to just flow where they want to flow. Sometimes we want to go against the flow. Sometimes we want to get a particular molecule and we want to concentrate that molecule. We want to get more and more and more of those molecules in the same spot. But that goes against the idea that they want to spread out against diffusion. And so the only way to fight against that and to start to concentrate the molecules is to use energy. And so there's a lot of different ways that this can happen. For example, in this particular one, this is a pump that is taking the orange shapes and pushing them in, and the yellow shapes and pushing them out. The idea being that there's already a bunch of orange shapes on the inside, but we want even more to get in there. And so we have to kind of force those to go in there. Meanwhile, on the passive side, you could see this being something like carbon dioxide or oxygen molecules, which are easily can pass through the cell membrane because they are small and non-polar, and they're just going to go from high to low. Here we have a molecule that's going to go from high to low, but maybe this one's a little bit bigger. Maybe this one is like water, or even maybe glucose, and we want to just make a tunnel that's going to let the diffusion work on its own. There's no energy necessary because it's still going from high to low, but we needed to help it. And that's where this word called facilitated comes in. Facilitated diffusion is still passive transport because it's using the concentration gradient, but we had to help it somehow or another to let that go. One big form of active transport is called endo and exocytosis. This would be for molecules and particles that are just bigger, too big to fit through some little tunnel that we want to make. And so in this case, we're going to have the cell membrane start to change its shape a little bit. For example, in endocytosis, which is being shown in this part right here, or in this part right here, you can see that the cell membrane starts by making like a little U shape, right? It's actually bending itself, and then it's going to bend to make that into a circle. It's that U shape is going to push on both sides to make a circle, and then that little circle is going to pinch off and become this little bubble that we call a vesicle, right? And of course, that's going to take energy because the cell membrane isn't going to just start like moving itself around without any energy to make it do that. Exocytosis is the opposite, where the little bubble fuses with the cell membrane, as kind of being shown in this particular animation right here, where the bubble is just kind of fusing with the membrane, and anything that was inside that bubble is about to get pushed to the outside of the cell. And again, for endocytosis, you just imagine that animation going backwards, and you would have endocytosis, right? And so various different mechanisms to get stuff into and out of the cell. But there's one last part that really is important for understanding how the cell works, and that's the cell size. That the getting things in and out of the cell quickly is important. You want to be able to respond to the environment as quickly as you can. And so because most of this going on inside of the cell is based on diffusion, it's based on just a slow flow of molecules from one place to the next. This is only going to work best on short distances. Sorry about that. And so what we want to get is a lot of surface area. We want surface area because that's where we're going to have most of our proteins, and this is where we're going to get a lot of the work done. But the way to get more surface area is not to make the cell bigger. Of course, making the cell bigger does increase the surface area. In this particular case, we have the surface area of 6 here compared to 150. So definitely the surface area went up, but not nearly as much as the volume went up. The volume went from 1 to 125, and that's a much bigger change. And so the idea is that volume increases way too fast. As you make the cell bigger, volume increases really fast, surface area only gets bigger, not quite as fast, and you end up having a problem. The idea is that we want the most surface area compared to volume. And so here's where I had these marks left over from last time. But if you notice in the big block compared to lots of little blocks, the volume is the same, but the surface area is much, much higher when you have a lot of little blocks, right? So we want to have a lot of small cells rather than a few big cells, or else transporting things in and out starts to become way too slow, and then the cell's not going to be able to survive, right? Now, again, that's all based off of diffusion. That diffusion is going to be what the cell's going to utilize as much as possible. We don't want to use energy as often as we can. We don't want to use energy. We want to save that energy, right? So diffusion is something that we need to talk about quickly and remind ourselves that diffusion is the movement of molecules from high to low concentration. So if I were to drop some sort of red food coloring into a beaker of water, there would be a lot of red food coloring there, but there would be low concentrations everywhere else. And we would watch as the food coloring would spread out through the beaker of water. Now, that's the basic rule, is that molecules are going to go from high to low concentration. But the important part is this is based on motion of the particles, right? That particles are always vibrating and bouncing around in their little mosh pit. And so as they slam into each other, this is going to force them farther and farther apart. This animation is showing that the movement is never stopping. I'm going to keep going back and forth between this one. But that while most of the particles are moving left to right, there are still some that are moving right to left. There are some that start out moving from the left to the right, but then they get collided with and start to move back the other way, right? There's all kinds of chaos going on down there. But the basic idea of the chaos is that the molecules are never going to stop, and that when we get to equilibrium, everything's going to be moving both in and out of the cell as fast as you can because of the constant motion of the molecules. And so if the molecules are constantly moving, they're going to want to spread out and come to equilibrium. But this is hard when you have a semi-permeable membrane. because in a semi-permeable membrane, certain things can't go across the cell membrane, such as, in this example, sugar. And so while you have a lot of sugar over here in the original scenario, it would want to move to the left, but it can't because the barrier is in the way, the membrane's in the way. So instead, the water is going to end up being the thing that moves, because the water often is what can always diffuse wherever it needs to diffuse. And so this is a special situation of diffusion called osmosis. And so osmosis is very important for the health of a cell, because we need to maintain our water balance all the time inside of our cells. And that leads us to these scenarios that hopefully you remember. You have the solutions that are either hypotonic, isotonic, or hypertonic. We want to know where is the concentration gradient and where is the water going to flow. Now, on all these situations, the tonic is the solute. It's the stuff that is dissolved in the water. And then the prefix hypo means low, and then iso means equal, and hyper means high. So when we look at the first example over here of the hypotonic solution, that's telling us that the solution has a low amount of stuff, low solute. This then means that it has high amounts of water. And if there's high amounts of water on the outside compared to the inside of the cell, then as you can see in the arrow, the water is going to flow into the cell. On the opposite situation, in a hypertonic solution, the solution on the outside of the cell has a high amount of solute, and that means it has a low amount of water, and so the water is going to go from the inside of the cell towards the outside of the cell because there's a low amount of water on the outside of the cell. Again, maybe the other way to remember this is that the water is going to go where the stuff is. So in a hypertonic solution, the stuff is on the outside, so water goes on the outside. In a hypotonic solution, there's a lot of water on the outside, which means there's a lot more stuff on the inside, and so the water is still going to go to the inside where there's a lot more stuff. The isotonic situation, of course, has two arrows because there's water going in and out, just kind of diffusing randomly, just based on the movements, right? Now, this particular graphic does have two parts to it because there's the animal cell and the plant cell. And while the hypo and hyper and isotonic solutions have the same arrows, you might notice the arrows are the same. Because plant cells have a cell wall, this is going to make the consequences different. In this particular example, where you have too much water on the outside of the cell, the water is going to want to come in. And if you're a human, this is going to make your cells burst. This is something that unfortunately doctors learned way back in the day when they first started to try to do transfusions, and they thought, hey, this person lost a lot of blood, let's put a bunch of water into their blood. And that just made their cells explode, which is a bad thing if you want to live. But if you're a plant, this is exactly what you want, because the plant relies on that water that is constantly trying to squeeze its way in to provide pressure, and that pressure is what keeps the plant upright. Over here on the right-hand side, when you have the hypertonic solution, the water is going to flow out just like in the animal cell, but the cell wall is going to remain. And so the cell wall provides some amount of structure, but now all that water that was supporting all the weight of the plant is gone. And so the plant is going to wilt. It's going to fall over in this situation. Of course, you can reverse that by trying to water your plant and adding more of a hypotonic solution. And then the water will start to flood back into the cell, and the plant will hopefully right itself back up if it hasn't suffered too much damage from being wilted before that. Humans, of course, want the isotonic solution. That's what we would like. Eventually, doctors figured out that you shouldn't just put pure water into somebody's body. You should put saline solution, which is basically just salt water that happens to match the concentration of the solutes in our blood. And then when you give that to somebody, your body just is fine with it because it's exactly the same as before. Oh, here's this slide right here that shouldn't be here yet. Or should it be here? Yes, it should be here. And I just gave the answers away. But here's something that we would do quite often is we want to know what is going to happen when we have the cell. Where's the water going to go? And so you have your cell, you have your solution outside the cell, and we want to start to look at the concentrations. Now, in this particular example, we're going to assume that the membrane is not permeable to sucrose and glucose and fructose. So only the water can move. So as I already have let on here, on the left-hand side, there's 0.05 molar concentration of solute and only a 0.03 in the environment. And so when we're looking at the cell compared to the beaker, the cell is going to be hypertonic because there's more stuff. There's 0.05 molar inside the cell. We could describe the solution, which would be a hypotonic solution. But in the end, we want to know where the water is going to go. And again, water is always going to go from where there's a lot of water to where there's a little bit of water. Another way of thinking of that would be that the water is going to go where there's more stuff. So the water is going to go into the cell, and that cell should start to swell up in size as the water flows in. There is some math that goes along with this called water potential, but I'm not going to try to put that onto this video. There'll probably be its own video for that. But there you have the basics of the cell membrane, their structure, their function, and then transport, including diffusion. As always, let me know if you have any questions.